To date, we are distributed in 108 countries. Just considering last year, we sold about 130 million tests in the world. Alifax Test 1 and Alifax Roller 20, using quantitative capillary photometry, patented and invented a new method to get ESR, erythrocyte sedimentation rate, in 20 seconds. The reference method to measure ESR is Westergren. That takes 1 hour for a single sample of blood. The ESR test is a simple blood test that helps to detect inflammation in the body, measuring the sedimentation of red cells. U.S. Patent Office accepted our patent. This is a representation of photometry detection during the stopped and flow procedure. So Alifax Company was free and authorized to present to the world the new ESR, correlated with the Westergren method. Alifax is the only one company in the world using quantitative capillary photometry techniques to measure ESR. Alifax, for the first time in the world, shortened the ESR test from 1 hour of Westergren to 20 seconds. For the first time, Alifax eliminated the need of dedicated tubes for the blood collection, using the same hematology tubes used for other tests. In the medical and diagnostic business, Alifax has substituted the use of dedicated tubes for collecting the blood sample for the ESR, with hematology tubes also used for other tests. Alifax, for the first time in the world, has introduced the use of smart cards to run thousands of tests without producing plastic tubes to be disposed of. The smart card and the technique used by Alifax significantly reduce the stock of dedicated plastic tubes. To simulate the flow of blood inside the instrument, Alifax used a capillary tubing of 0.8 millimeters internal diameter. The capillary tubing is heated at 98.6 Fahrenheit, 37 Celsius degrees, to simulate the human body temperature. The roller uses patent technology for instruments and applications in order to overcome all the problems and limitations of Westergren method, as described from the American Guidelines documents, Clinical and Laboratory Standards Institute, CLSI. We designed a super reliable method in 20 seconds instead of 1 hour of Westergren. As a result, more than 9,000 instruments work around the world. In detail, the needle enters inside the blood test tube, and, using a peristaltic pump, about 30 or 170 microliters of blood are drawn and pushed into the capillary tube. After a few seconds, the pump stops, and due to the inflammation status, the erythrocytes aggregate with particular proteins named agglomerins. These aggregations of red cells and agglomerins are called rouleaux. When the flow stops, two detector photodiodes measure the level of light, delivered from emitter sources, passing through the capillary tube. Each blood sample is monitored for about 10 seconds with 1,000 miserations. The variation of the light, kinetics, is correlated with the Westergren method. In simple words, if red cells aggregate due to the presence of agglomerins, the variation of light is high. On the contrary, if no aggregation happens, the variation of light is low. Alifax has created a unique and specific selectogram for the red blood cell sedimentation rate. During the experimental studies and methods setup, thousands of blood tests were performed to obtain the results we are showing you today. As for the Westergren test, Alifax ESR test is in millimeters per hour. We have more than 50 scientific publications with our method. We obtained recognition for our ESR method that provides a result in 20 seconds. And just to show two of these, the University of South Korea declares that Alifax technology is better than Westergren method for detecting inflammatory status of patients. Alifax technology is better than the reference method. The Wall Street Journal in 2012 declares that there are approximately 75 to 80 million ESR tests ordered annually in the US. To date, Alifax is the only company in the world that offers the ESR in 20 seconds. Leading innovation forever, and thank you for your attention.
Intelligent instrument management is at the heart of a well-maintained automated laboratory. Users can control the readiness of the V8 Nexus to match your workflow, ensuring that when you're ready to analyze samples, so is your instrument. Starting up the V8 Nexus couldn't be simpler. The dual power controls allow the instrument to be intelligently managed, controlled through our unique Platinum touchscreen software, used for setting auto startup, shutdown, and idle times. Selecting the default test mode ensures the V8 Nexus is configured to accept testing samples correctly. It's important to ensure the appropriate buffers, waste containers, and consumables have all been loaded prior to startup. By incorporating this into a daily routine, the V8 Nexus will always be ready to accept samples. To find out more, visit our website. Ordering tests for analysis by the Helena Biosciences V8 Nexus is simple to carry out. When preparing your V8 Nexus for startup, a default test will be selected. Ordering individual tests that differ from the default method can be done easily and all still within the fully comprehensive audit trail. By using the drop-down menu, it is easy to select your testing method. Ordered tests will appear on the left-hand side of the screen in the ordered tests list. Once precondition is complete, samples are ready to be accepted onto the system. Simply load the sample racks and place in the sample handling bay. The V8 Nexus will do the rest. There are many configurations for operators to consider when ordering tests. To learn more, visit our website.